Let's take a look at translation. In translation, the messenger RNA codons are going to be translated or decoded into the correct amino acid, which will then be joined together by peptide bonds to form a protein. And translation, we're going to be using both transfer RNA or tRNA and ribosomes. The transfer RNA is going to be the translator. So it's going to be the decoder, and then our ribosomes are the protein-making machines. And remember that translation is going to occur in the cytoplasm. So here's just a picture again of our eukaryotic cell, just reminding you that translation is going to be occurring in the cytoplasm because that's where the ribosomes are. So the transfer RNA, the reason they call it that is because it's going to transfer the amino acids to the ribosomes to add to the growing protein. And the transfer RNA structure contains an anticodon, which is just a sequence of 3 nucleotides that are going to be complementary to the messenger RNA codon. And then each transfer RNA is going to be bound by a specific amino acid. So let's just show you how this works. So if we have our DNA template triplet, so here we have GAA on our DNA template strand. And then when we transcribe our DNA template strand, our messenger RNA will end up being CUU. So you can see the G and C and A's and U's match up. And then the transfer RNA will come in and bind to the messenger RNA codon. So our transfer RNA anticodon will be complementary to the messenger RNA codon. So you can see G's and C's and A's and U's match up here. And then this transfer RNA will be bound to the amino acid leucine. Take a look at the structure of our transfer RNA. They have kind of an interesting shape. So they're kind of like a clover leaf. And you can see at the one end, they have the amino acid bound to them. So here we've got the leucine bound. And then at the other end, here's the anticodon. So you can see GAA is their anticodon. And then here's the messenger RNA that they're going to be matching up with the codon here, CUU. So you can see that they're complementary to each other. The G's and C's and A's and U's match up. So here's our genetic code table. And with every organism that we've looked at so far, everyone uses the same genetic code. So that means that our genetic code is universal, and whatever cell that gave rise to all living organisms that are alive today use the same genetic code, and it's just been passed on to everyone else that, all organisms that are alive today, so everyone still uses it. So there's 64 different codons here. 61 of them code for amino acids, and then we have 3 of them that are stop codons. Sometimes they're also called nonsense codons, but these stop codons actually don't code for any amino acids. So these just say that, hey, that's the end of our protein, stop translating. The other thing to notice about the genetic code is that it's what we call degenerate, which means that it's redundant. So you can see that a lot of our amino acids here, there's 20 amino acids, but there's actually 61 messenger RNA codons here. So the numbers do not line up. And what we mean is that there's a lot of these codons that are going to be coding for the same amino acids. So you can see in this bottom left corner, we've got 5 messenger RNA codons here that are all coding for the amino acid valine. So that's pretty common. that we do have several messenger RNA codons all coding for the same amino acids. So there is a lot of redundancy in the genetic code, and that's how we can get these silent mutations. So you can see that if you add the messenger RNA codon GUU and then it got changed to GUC, you would still be coding for valine. So that would cause a silent mutation. Okay, so how you use this genetic code table. So let's say that you had the messenger RNA codon of CUU, like we had on our last slide there. So you would take your first letter, we have C, and the first letter is always going to be on the left-hand side, and you just go along and you find your C. So here's our C right here. So we know that we're going to be in this block in this row here. And then your second letter is a U. And so your second messenger RNA letter is going to be up here, and you look for the U, and that would be here. So it would be this column, this first column here. So we know that we've got a C and a U. So we've got those two. And then your third letter is going to be this right-hand side over here. And so if you know that you're in this this second row, first column, then you know that, oh, you've got to be this U here. And so you go over and you're like, oh, there's the one I'm looking for, CUU. And you can see that that codes for the amino acid leucine. So that's how you use this genetic code table. All right, so let's just take a look at how translation works. So translation initiation, or starting translation, the ribosome is going to attach to the messenger RNA, and it's going to look for the start codon. So the start codon is always AUG, and that's always going to code for the amino acid methionine. So whenever you make a protein, the first amino acid is always going to be methionine. So because of that start codon being AUG, coding for methionine. So the ribosomes are actually in two parts. So this small subunit of the ribosome will bind first to the messenger RNA. We'll fit our transfer RNA onto the start codon with the coding for the methionine. And then we'll fit the large ribosomal subunit over that, and then we'll be ready to start translation. Okay, and then the ribosome is just going to do this cycle until it gets to the stop codon. So we're going to bring in our next transfer RNA with our next amino acid. It's going to come in and match up with the next messenger RNA codon. So its anticodon will match with the messenger RNA codon. And then we'll form a peptide bond to join this new amino acid to the growing protein chain here. And then once we've got that joined together, then the ribosome will move down to the next messenger RNA codon. Our empty transfer RNA will exit the ribosome, and now the next transfer RNA will come in and bring our next amino acid, and then we'll just start over and then join that new amino acid to our growing protein chain and keep going until we get to our stop codon. Okay, and then once we hit one of the three stop codons, you don't need to know what the stop codons are, but once we hit a stop codon, that's just going to tell the ribosome that that's the end of the protein. we can stop translating. We don't need to add any more amino acids. So the stop codons actually don't code for amino acid. What will happen is a release factor will come into the ribosome, and that will make everything fall apart. So the protein gets released and the ribosome falls apart, and now your protein will be ready to use.